Immunity. Every day, our body is exposed to many diseases, but still we are affected by few diseases because of our immune system, which protects us from many harmful diseases. Without an immune system, we cannot survive in a normal environment. Immunity is of two types: innate immunity and two, acquired immunity. Innate immunity. Innate, natural immunity is so named because it is present at birth and does not have to be learned through exposure to an invader. It thus provides an immediate response to foreign invaders. However, its components treat all foreign invaders in much the same way. They recognize only a limited number of identifying substances, antigens, on foreign invaders. However, these antigens are present on many different invaders. Innate immunity, unlike acquired immunity, has no memory of the encounters, does not remember specific foreign antigens, and does not provide any ongoing protection against future infection. The main components of the innate immune system are 1. Physical epithelial barriers, 2. Phagocytic leukocytes, 3. Dendritic cells, 4. A special type of lymphocyte called a natural killer NK cell, and 5. Circulating plasma proteins. Acquired immunity. The acquired immune system evolved in early vertebrates and allows for a stronger immune response as well as immunological memory, where each pathogen is remembered by a signature antigen. Antigen specificity allows for the generation of responses that are tailored to specific pathogens or pathogen-infected cells. The ability to mount these tailored responses is maintained in the body by memory cells. Should a pathogen infect the body more than once, these specific memory cells are used to quickly eliminate it. T cells. There are several types of T cells. The most important are killer T cells, helper T cells, and gamma T cells. Killer T cells directly attack other cells carrying foreign or abnormal antigens on their surfaces by releasing cytotoxins and a protease, granulysin, to target cells to undergo apoptosis. T cell killing of host cells is particularly important in preventing the replication of viruses. Helper T cells regulate both the innate and adaptive immune responses and help determine which immune responses the body makes to a particular pathogen. These cells have no cytotoxic activity and do not kill infected cells or clear pathogens directly. They instead control the immune response by directing other cells to perform these tasks. Gamma T cells possess an alternative T cell receptor, TCR, and share the characteristics of helper T cells, cytotoxic T cells, and NK cells. Gamma T cells straddle the border between innate and adaptive immunity. B cells. A B cell identifies pathogens when antibodies on its surface bind to a specific foreign antigen. An antibody is made up of two heavy chains and two light chains. The unique variable region allows an antibody to recognize its matching antigen. This antigen antibody complex is taken up by the B cell and processed by proteolysis into peptides. The B cell then displays these antigen peptides on its surface MHC class II molecules. This combination of MHC and antigen attracts a matching helper T cell which releases lymphokines and activates the B cell. As the activated B cell then begins to divide, its offspring, plasma cells, secrete millions of copies of the antibody that recognizes this antigen. Memory. When B cells and T cells are activated and begin to replicate, some of their offspring become long-lived memory cells. Throughout the lifetime of an animal, these memory cells remember each specific pathogen encountered and can mount a strong response if the pathogen is detected again. Immunological memory can be in the form of either passive short-term memory or active long-term memory. Passive memory is especially important for newborn infants which have no prior exposure to microbes and are particularly vulnerable to infection. During pregnancy, a particular type of antibody called IgG is transported from mother to baby directly across the placenta. Active immunity. Active immunity. When a host is exposed to the living or dead microbes or protein, antibodies are produced in the body. Active immunity can also be generated artificially through vaccination. Passive immunity. When ready-made antigens are given directly to protect the body. The principle behind vaccination, also called immunization, is to introduce an antigen from a pathogen in order to stimulate the immune system and develop specific immunity against that particular pathogen without causing disease associated with that organism.